Hello, and thank you for joining us today for the CITI program's webinar, Ethics and Policy Issues in CRISPR Gene Editing, presented by Josephine Johnston. My name is Jamie Arango, and I am the Director of Educational Initiatives at the CITI program, and it's my pleasure to welcome you. I'll be serving as your moderator today. So let me tell you about today's presenter, Ms. Josephine Johnston. Ms. Johnston works on the ethics of emerging biotechnologies, particularly in human reproduction, psychiatry, genetics, and neuroscience, and is the Director of Research at the Hastings Center. Her scholarly work appears in medical, scientific, policy, law, and bioethics journals. She is co-editor of Human Flourishing in an Age of Gene Editing and Trust and Integrity in Biomedical Research, the Case of Financial Conflicts of Interest. Ms. Johnston holds degrees in law and bioethics from the University of Otago in New Zealand.
We're going to talk today about a new technology that I'm sure everyone has at least heard of, and some of you may know a lot. I think it's even possible there are people on the webinar who know a lot more about the science of this technology than I do. My background is in law and bioethics, and so I'm really taking a policy and ethics perspective on this technology, and there's plenty to say about what's been happening in those realms as well as scientific advances. So we'll try to cover as much of that as we can today. I want to note that the emphasis today is going to be on the use of this technology, CRISPR gene editing tool, in humans or in the preclinical work that could lead to use in humans. CRISPR is a gene editing tool that allows for the addition, removal, or alteration of DNA, include in many kinds of different cells, actually, and just in DNA generally, but it also can be used in human cells. It has greatly accelerated research and has potential uses in a variety of organisms, as I said, all the way from mosquitoes to human beings. So there's a lot to be said, and we are really going to focus on international and US policy and ethics responses to the technology and its use in humans. And we'll compare that to some ethics and policy responses that have occurred before CRISPR, because there's a lot of there are a lot of connections between what we're talking about today and what's been discussed for decades actually in the past. Here's our learning objectives slide for this work. It's really, I've divided into 6 different areas. We'll talk a little about the emergence of CRISPR, some of the previous technologies, ethics and policy issues, what's been going on in the realm of international governance. We'll look at US policy for human gene editing. We'll specifically look at the case of germline gene editing, which has received a significant amount of the attention's realm, and we'll think a little bit about future questions and directions, so things to keep an eye on going forward. This is not a resolved area of science, ethics, or policy, and so there's a lot of moving parts here, so we'll think a little bit about where things might be headed. Here I've put up just a few headlines from 2015 and 16, which was when the mainstream media really caught on to the significance of this discovery as it became more and more clear that this really could be a tool for making changes to the human genome. So in 2015, Wired had a headline on the cover of its magazine, "No hunger, no pollution, no disease, and the end of life as we know it, the Genesis engine." So very significant sounding headline. MIT Technology Review, "We can now engineer the human race." So very quickly, the implications for use in humans were big news. And then Time magazine, so very much mainstream journalism, "The Gene Machine, What the CRISPR Experiments Mean for Humanity." When you talk to scientists working in the field, CRISPR is of course a very big deal because it really has accelerated their research, but they will point out that it's very much related to research that's been going on for decades. So it's both a very big deal and also just a new tool for doing things that we've been trying to do for a long time. In the US, there was a policy response to the recombinant DNA discussions, which was the creation of an advisory committee at the NIH to look at research involving recombinant DNA, development of biosafety rules relevant to that area, and of course not sparked actually by anything related to gene transfer research or genetic engineering, but there are long-standing rules and regulations, of course, around human subjects research, around FDA's oversight of clinical trials, that all form the background or a backdrop against which CRISPR lands. At the international level, There are a couple of conventions or declarations that mention the human genome and inform the way that we think about CRISPR today. Some of those, I've quoted a couple of them here. Neither of them says that you can't make changes to the human genome, but they do call attention to the human genome as a significant entity, I guess, in itself. So the Universal Declaration on the Human Genome and Human Rights says that the human genome underlies the fundamental unity of all members of the human family, as well as the recognition of their inherent dignity and diversity, and is a symbolic, in a symbolic sense, is the heritage of all humanity. So you can quibble with those words, but the intention there is certainly to signify that the human genome is something of particular significance to all of us. And similarly, the Universal Declaration on Bioethics and Human Rights from 2005 really calls attention to future generations and their genetic constitution, and asks us to pay attention to the future generations in our discussions today. So both of those really kind of, I think, inform how we think about CRISPR. And then at the level of national laws, the US has not done this, but a number of nations in the 90s and subsequent to that passed laws that prohibited altering the DNA of egg, sperm, or embryos. So essentially prohibiting creating new future persons with altered DNA. So those, of course, come in to be. In addition to these international sort of high-level activities, and there are a couple more that have subsequently happened that I'll tell you about in a second, I want to also just note that the US has had its own policy response. So as I said before, there were pre-CRISPR, of course, a lot of different rules and laws that have actually some implications for CRISPR. One of them is the Dickey-Wicker Amendment, which is a piece of legislation that's been part of budget acts for NIH since 1996 that prevents the use of federal money for research that might involve destroying or harming a human embryo. Now, we all focused on this a lot during the early days of the embryonic stem cell research controversy, but this Dickey-Wicker Amendment would also impact any researcher who wanted to do research that could, looking at germline modification of embryos, because you would not be able to use NIH monies for that. There are, of course, ways to use other funds. NIH has had a policy since 2015. It's made a couple of policy statements. Francis Collins, as the head of NIH, has made these statements that it will not support research to make on germline modification. So even aside from the use of issue about the use of embryos, they're not looking for germline modification research and they not want to fund it. And of course, the RAC already existed as a body to look at this kind of work. Post-CRISPR, the NIH, as I said, has reiterated its anti-germline modification position twice. They've revised the RAC and turned it into a new kind of committee that is called the Novel and Exceptional Technology and Research Advisory Committee. So slightly broader remit, but certainly to look at the ethics of this kind of technology. Perhaps the most significant post-CRISPR piece of US policy is an FDA appropriations rider that has been attached to budget bills since 2015. So it came out for the first time a couple of weeks after the International Summit, or maybe a week after it ended in December 2015. This rider prohibits the Food and Drug Administration from using federal funds to review research in which a human embryo is intentionally created or modified to include a heritable genetic modification. So what this does is it prevents the FDA from even looking at a clinical trial involving heritable modification, and that is essentially how we currently have a situation where the kind of research that He Jiankui did could not be done in the United States, because you would need FDA approval to move forward with it, and they will not and cannot look at it.
I want to thank you, Josephine, for today's presentation. Also want to say thank you to our audience members for joining us today and for their questions.